Hello, my name is Mansi Vora. Today I'm presenting the cell cycle, cell signaling, and their relationships to disease, including cancer. The cell cycle is an ordered series of events involving cell growth and cell division that produce two new daughter cells. There are two phases. First is the interphase, and the second is the mitotic phase. Interphase is a cell are preparing for the cell divisions. During this phase, also include some stages G1 phase, S phase, and G2 phase. Then after they move from interphase to the mitotic phase. The three phase is a G1 phase is the cells are growth in this phase. In a S phase, cells are growth and also DNA synthesis. During the G2 phase, the final preparation for the cell divisions. Then mitotic phase. The mitotic phase is a multi-step process during which the duplicate chromosomes are eliminated or separated and moved to opposite poles of the cells, and then the cell is divided into two new identical daughter cells. The first portion of the mitotic phase, mitosis, is composed of five stages, which accomplish the nuclear divisions. The second portion of the mitotic phase, called the cytokinesis, is the physical separation of the cytoplasmic components into two new daughter cells. The mitosis is divided into a series of phases, prophases, promoptophases, metaphases, anaphases, and telophases. In the result, in the division of the cell nucleus. Here is a table which is all information about the series of the phases. Now the control of the regulation of the cell cycle. The cell cycle is a tightly controlled and regulated by proteins called the cyclin and cyclin dependent kinases. Here are certain checkpoints. G1 checkpoints is a G1 checkpoints is a determines whether all conditions are favorable for a cell divisions to produce the G1 checkpoints, also called the registration points for the at which the cells irreversibly commits to the cell process. The G2 checkpoints the G2 checkpoints bars the entry to the mitotic phase of if a certain condition are not met as in the G1 checkpoint, cell size and proteins results are assessed. During the M checkpoints, the M checkpoints occurs near the end of the beta phase stage of the mitosis. The M checkpoints is also known as the spindle checkpoints because it determines if all the sister chromatids are currently attached to the spindle microvated. Now the cytokinesis. Cytokinesis is the second part of the mitotic phase during which cell divisions is completed by the physical separation of the cytoplasmic components into two new daughter cells. Now the changes in a gene expressions. The mammalian cells requires activation of at least two cyclin CDK complexes. First is cyclin CDK4 and the second is the cyclin E CDK2. Here is the activation of cyclin CDK complexes. The cycle begins with the binding of CDK2 cyclin. A quaternary complex is formed. This stage is marked as a inactive. Both activing and inactive kinases of phosphorylate CDK. The complex remains inactive in this stage. CDK inactive phosphate removes the immediately phosphate activating the complex. The process continues with CDK inhibitors binding to active on inactive complexes and preventing the complex from functioning. Now, the relevance of disease. The cell cycle is disrupted. It can lead to a number of disease, including cancer, cardiovascular disease, and neurodegenerative disorders. Now, the cell signaling, a biological mechanism that occurs in a cells, which gives cells an ability to receive, generate the signals in a response to their surrounding environment. The cell-to-cell communication between cell is mediated by extracellular signal molecules. Some of those operate over long distance, other signals on only to immediate neighboring cells. There are types of cell signalings. These are types: direct, paracrines, and endocrines. The direct signaling is the cell must be direct contact. Second is a paracrine signaling signal that act locally between cells that are close together are called paracrine signals. The endocrine signals from a distant cell are called endocrine. Now the steps of the cell signalings. There are three steps: receptions, transductions, and response. The reception cells detect the signaling molecule from the outside of the cell. A signal is detected when the chemical signals, also known as a ligand, bind to a receptor protein on the surface of the cell or inside the cell. The transduction, when the signaling molecule bind the receptor, it changes the receptor protein in some way. This change initiates the process of transduction signal. Transduction is usually a pathway of several steps. Each rarely molecular in the signal transduction pathway changes the next molecular in the pathway. The third is a response. It's a finally the single triggers a specific cellular response. Now the ligands. Ligands which are produced by signaling cells and interact with a receptor in or in a target cell come in a many different varieties. Some are proteins, other are hydrophobic molecular like steroid, and other are gases like nitric oxide. The types of ligands. First is a membrane impermeable. Second is a membrane permeable, and third is a physical signals, pressure, temperature, light. Now the cancer disease. Cancer is a physical disease of uncontrolled cell divisions. They behave differently than a normal cells in the body. Many of these differences are related to cell division behavior. Here we show the difference between the normal cells and cancer cells. The cancer cells are also different from a normal cells. In another way, they are not directly cell cycle related. This is the different help them grow, divide, and from tumors. From instance, cancer cells gain the ability to migrate to other parts of the body, a process called metastasis, and to promote the growth of new blood vessels, a process called agenogenesis. Now the tumor suppositories. Negative regulators of the cell cycle may be less active in a cancer cell. For instance, a protein that helps cell cycle projections in a response to DNA damage may no longer sense damage or trigger a response. Genes that normally block cells to progression are known as a tumor suppressors. One of the most important tumor suppressors is a tumor protein P53, which plays a key role in a cellular responses to a DNA damage. P53 acts primarily at a checkpoint where it blocks cell cycle progressions in a response to damaged DNA and the unfavorable conditions. When a cell DNA is damaged, sensor protein activates P53, which helps the cell cycle as a checkpoint by triggering production of the cell cycle inhibitor. This supports buying time for a DNA repair, which also depends on a P53. Was the second job is to activate a DNA repair enzyme. If the damage is fixed, 53 will release the cell, also allowing it to continue through the cell cycle. If the damage is not fixable, P53 will play its third and final role. Is it triggering properties so that damaged DNA is not passed on. Now the difference between oncogenes and the proto-oncogenes. The positive cell cycle regulator may be overactive in a cancer. For instance, a growth factor receptor may second signal even when growth factor are not there, or a cyclin may be expressed at a abnormally high level. The overactive forms of these genes are called the oncogenes, while the normal, not at a muted forms, are called the proto-oncogenes. Now the cell signaling for the cancer. Cell signaling is the process by which cell receive and process information to respond to their environment. When cell signaling goes wrong, it can lead to a cancer. Dysregulation in a cancer includes first is the oncogene activation, example RAS, PI3K. Second is the loss of negative regulation, example PTEN, mTOR. Third is the overactive receptor, example is EGGR and HER2. The RAS, RAF, MAP kinase pathway is a upstream component of the pathway such as the RAS and RAF are protect oncogenes in a cooperation with other events can lead to profound changes and also transforming normal cells into fully malignant. Here is a pathway diagram. The components of this pathway are under intense investigation as a possible target for anti-cancer therapeutics. Mutated RAS is associated with 20 to 30% of all human cancer are often not responsive to established therapies, resulting in a staggering 3 million new cancer diagnoses worldwide each year with RAS mutation. Now PI3K AKT mTOR pathway. This pathway is an intercellular signaling pathway important in appropriate enhanced cancer, example of breast cancer and non-small cell lung cancer and longevity. This pathway is activated by IGF-1 and has a number of downstream effects which either promote protein synthesis or inhibition protein breakdown. In many cancers, this pathway is overactive, thus reducing apoptosis and allowing proliferation. The cancer drugs trials aim to inhibit the signaling sequence at some point. Now, the cancer treatment therapeutics to traditional to modern approaches. There are many therapies, exomer-based therapy, stem cell therapy, gene therapy, nanoparticle-based therapy, and also natural product therapy. So first is exosome-based therapy, the tumor-release exosomes, T lymphocyte cell-divided exomers, M lymphoma cell-divided exomer, natural killer cell-divided exomers, and many others. In a stem cell therapy is also adult stem cells, cancer stem cells, gene therapy is a direct lysing tumor cells and introduce wild type tumors, suppressor genes into cells. Nanoparticle therapies, polymer-based nanocarriers, lipid-based nanocarriers, drug conjugates, viral nanoparticles, inorganic nanoparticles, and natural product therapies based on plant-based secondary metabolics. Here is my summary. The cell cycle is a process of cell division regulated by protein like cyclin and CDK with checkpoints, ensuring a proper DNA replication and division. Cell signaling controlled cellular activities through a pathway like MAPK and PI3K, responding to external signals via receptor. Disruption in this system can lead to disease, particularly cancer, where mutation in a tumor supports is example P53 or overactive signaling pathway like EGFR causes uncontrolled growth. The interplay between abnormal signaling and the cell cycle deregulation is a key driver of a cancer, making this pathway critical targets for therapies like a CDK and tyrosine kinase inhibitor. So here is the my presentation, and thank you so much.